Okay, so now we'll study the one more organelle. It is lysozyme. Lysozyme was first discovered by Alexander Fleming. There was again an interesting story behind that. Alexander Fleming, when he was working with the microbes and with the culture of the microbes, he had prepared some cultures with the tubes and plates, and he was working in his laboratory. And suddenly, while he was working, something get fall down from his nose. Okay, what is something that you know that get fall down into the culture of the bacteria, and next day when he observed those bacteria which he has cultivated, they were dead. He was surprised after looking that, because he has kept so many cultures at the same time, but the only one in which the mistake has done, or in which the incidence has done, he had marked it. Now we know the reason why it happened, because of the mucus of the nose, and he has started studying further on that, and he has gradually identified that there is something in such type of mucus things that kills the microbes and kills lice. Lice matlab todna. That lice, those things are called lysozymes. Okay, lice plus enzymes. Lice matlab todne wale enzymes. That's why he has given the name as lysozymes. Now what are lysozymes? The membrane bound sac filled with enzyme within the cell. From the Golgi apparatus, from the trans side, the smaller vesicles will get liberated. Those smaller vesicles that is filled with what? Enzymes, with the lysozyme. This sort of enzymes is found in tears, saliva, sweat glands and other mucoid lining. Mucoid lining in our intestine, in our saliva, in mucoid lining in our nose, in tears and in the sweat glands. It has got certain this type of lysozymes. Due to the presence of lysozymes in the tear, no bacteria can harm it. Okay, the same thing as with the skin. Skin has also got the lysozymes. That is why skin is protected from that. Understand? Okay, now we understand the function. What do the lysozymes do? Lysozymes are the bag-like structure that contains the enzyme. Okay, whenever a foreign material, or we can say the bacteria or viruses or whatever from the outside comes in our body, those are called the foreign material. Those foreign material, when get enter in our body, that will get killed by the lysozyme. Okay, the second one is when our cell get damaged, when our organelles, okay, when they get aged or damaged in our cell, within our cell, the cell organelle will get gradually digested by those lysozymes. Okay, the third important function is they are responsible for the killing of the whole cell when that cell is not functioning anymore, or when it is got dead or worn out cells. Okay, those cells which are old, those get killed by these lysozymes. Now this how this lysozyme will act on that? In the first and second mechanism, that is in the killing of the foreign material and during at the digestion of the cell organelle. What they do, I have tried to explain by this figure, that this is a mitochondria. Okay, this is our cell, eukaryotic cell. This is a mitochondria. Okay, this mitochondria, say for an example, it is run out. Run out matlab dysfunction, not functioning anymore. Now this mitochondria will around this mitochondria, the endosome will created. The vacuole-like structure will get created. This is the lysozyme. This lysozyme will come and fuse with the endosome. Okay, so this is the organelle. This organelle is worn out. is not functioning anymore, they will form the endosome. This endosome will fuse with the lysozyme. This and eventually this will digest whole this organelle inside, and that digested thing will thrown outside of the cell. Okay, this is the first function we talk about. In this mechanism, this is the bacterial cell. You can say this bacterial cell when get entered into the cell by phagocytosis, they will form, the cell will form phagosome. Vacuum like structure, they will create phagosome. And this lysozyme again will bind with will fuse with that bacteria and will break down or digest or kill this bacterial cells into the smaller part, break into the smaller part. Now this is a kind of waste material. This waste material now will thrown out, that is called exocytosis. Okay, endocytosis come in, exocytosis goes out from the cell. Okay, these are the two different phenomena. And the third phenomenon, this lysozyme will burst inside. This bag will burst inside the cell. So all this content from, there are not only one lysozyme present, there are multiple lysozymes in the cells. So they will get burst. When they get burst, this enzyme will get liberated within the cell. Because of that, all this content whichever present into the cell, they will get digested and the cell will eventually get killed. Okay, that is why this system is known as the waste disposal system, because it has disposed the waste, and also termed as the suicidal bag of the cell. Why? Because the cell will kill themselves with the help of this lysozyme. Okay, so these are termed as the suicidal bag of the cells. Now, see the structure over here. This is a ultra structure of lysozymes. This sort of lysozymes are present in the cell. It is an ultra structure. You need not to understand at your level as you are in 9th standard. Okay, so this kind of structures, they are present into the cell, and eventually they get burst, and this enzymes which is present within the cell, within the organelle, then that will get liberated in the cell and the cell will get died. Okay, so have a look on this structure. You will have better idea on with the help of this figure. Okay, how this bacteria get enter into the cell and get eventually digested by the lysozyme, and the waste material get thrown out. As well as in the second part over here, this lysozyme had acted on the organelle, thrown out organelle, and it will also get digested and thrown out by exocytosis out of the cell. Okay, you hope you understand this structure. Good. So the next cellular organelle we are going to study is mitochondria. Mitochondria has first find out by the scientist Albert von Keeler. Albert von Keeler, when he has seen, he told it as a bioblast, because there is something from which the continuous energy is generating. Okay, after some time, after some years, Richard Altman has now give the word term mitochondria to the bioblast. So first mitochondria was first find out by Albert von Keeler. Okay, but the term mitochondria was given by Richard Altman. Okay, there were two scientists involved in that. First invention, and the second one is the term mitochondria given by Richard Altman. Now, what is mitochondria? Mitochondria is the double membrane rod-shaped structure. This structure I have shown over here is a something like not complete round structure. It is with a rod-shaped like structure. This double membrane rod-shaped structure involved in energy generation through cellular respiration. As we know, when we breathe, we take oxygen. That oxygen will go to the cell level, and in the cell it will be absorbed by the mitochondria, and in the mitochondria, this cellular respiration will take place in the presence of sugar molecules. Now, whatever the energy received by the cell is in the form of ATP, adenosine triphosphate. Okay, this ATP is now termed as the energy currency. We can say our currency. If we want to buy something from the market, then we use the currency as a rupees. Over here, if we want to take some energy or we want to release some energy, ATP is the currency. When ATP will get break down, energy will get replaced. So whenever we need it, the ATP generation is a continuous process. But whenever we need it, it will be stored. And whenever we need it, this ATP will get broken down and energy get released. So these are the involved in the energy generation, and energy gets generated in the form of ATP. And ATP are the energy currency. Okay, now have a look on the structure. First I'll show the model. This is the structure of a model of mitochondria. As you can see over here, the cream color part, which is the outermost membrane. Okay, and this is the inner membrane which is folded inside the matrix. The red color part is the matrix. Matrix is nothing but the liquid portion of the cell. This second or inner membrane is inward into the cell matrix and termed as the cristae. Okay. So what is the function of the cristae? Cristae give the larger surface area for the generation of the ATP, because whatever the cellular respiration reaction takes place, that takes place on the surface of this cristae. So if the cell is completely raw, it will be a less space for the energy production. Now by doing this, they will make the larger surface area for the ribosomes to bind on them. Okay, and for the more energy generation, for the more ATP generation. Okay, so this is the structure. As this structure, if you see in the diagram, we can draw the diagram like this. This is the outermost membrane. This is the inner membrane. Inner membrane is folded inside, and that is termed as the cristae. Cristae gives the surface area for energy generation or energy generating reactions. Matrix, the fluid portion in the cell is called, in this organelle is called the matrix. Now the unique thing over here is this organelle has got its own DNA as well as its own ribosomes. This is the DNA I have shown, and these are the ribosomes. So with the help of their own DNA and own ribosomes, they can produce proteins. Okay, for the production of the protein, what we need, as we have discussed earlier, ribosomes. When both ribosomes and DNA. These ribosomes will go and bind with the DNA and produce the protein. So the mitochondria will produce their own proteins. It is quite easy. You can prepare your notes by your own and can write in the exam. Okay.